Now we've got a poll. This is a feature that we've really enjoyed getting the perspective from folks in the audience, and so we're going to put up a poll right now to start our first panel discussion. And here it comes. So the question is, are molecular methods like qPCR used to monitor your recreational water quality? So if you choose one, here they come. We'll take about 30 seconds or so to gather the data. All right. So it looks like we've got about a little over 60% of folks who are either using molecular methods or considering them, and about 37% that are not. And molecular methods are going to be a big part of the first panel. We're going to talk about that. As soon as we can get this moving. All right. So our panel session today is advances in monitoring approaches and technology. The session chairs are Rachel Noble. If you were following keeping score at home, you heard her mentioned in terms of this very topic, the monitoring technology. And we have Tracy Bone from US EPA as well as a session chair. So I'm going to hand over the helm to them, and we will get their panel as well.
Great, thank you so much, Steve. Tracy, I'm just checking sound. Can you hear me?
Yep, loud and clear.
Great, fantastic. So today we're going to be maybe pushing the envelope a little bit, talking in session 2 about advances in monitoring approaches and hopefully and technology, sorry, panel discussions. Our panel today is comprised of our 4 speakers in panel A. And those are Kendall Anderson, Oren Shanks, Laura Webb, and Shannon Briggs. If it's okay with you, Peter, I'll go ahead and embark on a short introduction of each of our panelists. Sound good?
Okay.
Can you hear me, Steve?
Can I go ahead? Sure can. Yep, absolutely.
Okay,
yes,
great.
You can.
Apologies, I thought I had lost you for a second. I'm in a completely different spot, so just double checking. So we're really pleased to introduce the speakers today. We have quite coverage over a range of different topics, covering not only recreational, what I would call routine monitoring in recreational waters, including Enterococcus and E. coli, but we also are covering topics today that really begin to get into sources of fecal contamination in the arena of microbial source tracking, as well as beginning to delve into thinking about cyanotoxins and harmful algal blooms. So first on our panel, in terms of order of speakers, Kendall Anderson is an environmental epidemiologist who specializes in microbiological and molecular biology for recreational and drinking water in sub-Saharan Africa. He's worked in the field since 2014 at Georgia Southern University, most recently in Chicago, Illinois, working for the Chicago Park District. And he's the project manager for beach water quality, in which he oversees the administration of the beach water quality program, and also working alongside with Drs. Abhilasha Shrestha and Sam Dorovich. And he's also begun to expand his roles in areas of remote sensing, which might be of interest to folks coming through in panel B for some of our other autonomous technology talks. Kendall has a master's in public health from Georgia Southern University, and also a master's of science from the University of Illinois at Chicago. When we think about our other speakers, we have Shannon Briggs, She has a PhD in pharmacology and toxicology, and she's a toxicologist with the Michigan Department of the Environment. I've known Shannon for a very long time, and she's an accomplished beach water quality advocate, but she's also undying in her ability to a desire to translate information to communities and to the public. She's a member of the Great Lakes Beach Association, and she's overseen in her tenure there at the Michigan Department of Environment increases in the use of molecular technology, multiple sets of intercalibration and molecular techniques, and she's really championed the training of laboratories in the context of microbial source tracking and sources of fecal contamination. She talks in her presentation about the method C, E. coli work, and she's recently expanded the lab network to 18 laboratories and digital droplet technology that they're interested in beginning to embark on. In addition, for our panel, we have Dr. Oren Shanks. He's a senior scientist for the Environmental Protection Agency in the Office of Research and Development in the Environmental Measurement and Modeling section. His primary research focus has been the development, validation, and implementation of molecular technologies, and he's been very much a champion of standardization of technologies and making those methods available across the country to counties, cities, utilities, and state laboratories. He's also been interested in fate and transport of nucleic acids, and he received his undergraduate and master's degree from the University of Wyoming and his PhD from Oregon State University. Last on our panel is Laura Webb. She's a chemist in the Field Services Branch, Laboratory Services and Applied Science Division of EPA Region 7. She's responsible for overseeing the water quality monitoring and operation of the accredited bacterial monitoring laboratories, but she's also been a major proponent of cyanotoxin monitoring and analysis strategies that she describes in her talk. So thank you for that opportunity to introduce our amazing panelists. Would you like to move on to questions, Steve?
That would be great. And thanks for those introductions. It really underscores the level of accomplishment and expertise and commitment of all the folks who are involved today.
Yeah, absolutely. So just making sure that I have this straight. So we're interested, at first I'll open up with some of the questions that the panelists have been kind of aware of. So I wanted to pose a question to Kendall first, and maybe we can move on to other panelists from there. You mentioned in your talk that it's taken some time to develop and prepare documents and tools that really convey to the public the timing and really the abstract nature of PCR results versus culture results, which as a community, as a research community and as a management community, we can agree that seeing colonies on a plate or seeing positive wells on an IDEX tray are far more kind of concrete results than PCR results that kind of come up on a computer screen. Kendall, could you talk a little bit more about that transition and some of the ways that you've tried to overcome these issues with the public and the media?
Sure, yeah. Can everyone hear me just fine?
Sure can. Thanks,
Okay,
Kendall.
good. Well, yeah, it has taken some time and taken a lot of adjustment over the years as we kind of get more advanced questions that tend to crop up from the public. But in general, when we began the process, we're taking some high-level research and then turning that into practice, but we also forget about taking these high-level concepts, and then you need to I don't want to say make them simpler, but make them provide analogies or something that people can connect with. A good one that I picked up while I was at university is comparing a thermocycler to a copy machine for DNA. And so making those comparisons to something the public can understand makes it a lot easier to communicate these difficult concepts. And really, when you start getting these questions and you start giving your answers, start writing them down over time and share that information with anybody that could potentially have contact with the public, because it works much better if you're all on the same page and you can communicate effectively as a team if you're in such a large organization such as the Chicago Park District. And really, anytime that you anticipate a change in your methods like we have coming up this year, you really need to sit down and think ahead of time. You're eventually going to get these questions, so sit down, think about them now, prepare statements that you can share with your press office even. So that'll really help instead of trying to scramble in the last minute to get something down.
And I realized this wasn't really part of the conversation or the prepared question, but how important is it also for you to make sure to convey to the public the limitations of molecular methods? Because all of the methods have limitations. Have you found that it's important to find balance there, or have you not gotten to that point yet?
That tends to come up with people that, let me rephrase. People that from the public that usually do have a more advanced knowledge of the methods, other scientists and maybe different fields that come to our board meetings and things like that, they'll have questions like that. And so you definitely do need to be prepared ahead of time. Probably the most common one that we deliver is that qPCR measures DNA. It doesn't matter if that DNA was alive or it was dead. So that is an important concept to address to the public when you get these questions too. And it's also if you have someone approaching you and asking, well, why did you switch from culture to qPCR? You need to be able to deliver the advantages and also delivers those disadvantages, and then to be able to speak with that person why those disadvantages are outweighed by the advantages of having something real-time qPCR based.
Right. So all of the panelists have worked in this space. Do you have any kind of advice or thoughts to offer, Shannon, Oren, Laura, on this transition? Because this kind of concept from a kind of the more traditional microbiology kind of pervades across all four of your talks.
Right. In Michigan, we use multiple methods. We still use co-alert, culture-based methods. There's still a place for that method. Some beaches are very, very far away from a laboratory, and rapid isn't going to make sense unless you can move a lab closer to the beach. So the co-alert test, that still works. You put your beaches on a schedule and you still use co-alert, not a problem. And for those beaches that are typically clean, co-alert works great. But there's some other beaches that, they're on and off, they're good, they're bad, and they fluctuate frequently. So you can't really tell what's going on unless you have a rapid test. So that's when the qPCR and the digital PCR methods have an advantage, that you can get a result faster. And there's even some other methods coming up that would give you a faster result. But then you have to keep in mind what's going on with all these methods is that we have a water quality standard in Michigan for culture-based methods. That's it. So we have to consider using co-alert as a water quality standard. So when anybody has, you're starting to approach a TMDL program, you will still be using most likely co-alert. But then on the flip side, once you've established a water body is impaired, then we would go after all the things that Oren Shanks has been teaching us with, HF 183 and the cow markers and all the source tracking. And it's not just qPCR, digital PCR, there's other things. People are using drones. I know Deneen has a presentation from your group, Rachel, that turned out pretty cool, a little boat. And I know Scorp has some of that. So it's really interesting to see all the different researchers coming up with different methods. So you got to keep in mind, what are your legal benchmarks? So for us, water quality standards using culture-based methods, but then there's a lot of other tools that you could use, and you do need to be aware of the limitations, but you can also use pretty much anything out of the toolbox and build your case, because you're usually going to have a community meeting, public meetings, and you're going to need to explain the source. So it's not just that figure out, is it meeting the standard or not? It's, if it is not meeting the standard, why? Where is it coming from? Why is this happening? And then we want to do the corrective action.
So still explaining it as a tool has intrinsic value really for what you do. You're using a battery of tools to try to solve a series of problems. It sounds like what's in common between your kind of thought process and Kendall's.
Right.
Yeah, that's great. I'd like to turn attention a little bit to microbial source tracking, Shannon, since you mentioned it. Oren, in your talk, you talked a lot about standardization, about providing standards, EPA methods 1696 and 1697, which, by the way, if others out there you know, aren't aware, a huge congratulations to you, Oren, for accomplishing this. I know it's been something that's been a long time coming, actually, since your graduate background, and for you to have gone all the way through the process and achieve this is really absolutely fantastic. So I think I can be joined in the rest of the both the research and the monitoring community in and saying this represents a huge amount of work, and thank you for it. Can you talk just a little bit about using those methods, however, in scenarios where you might see high bacterial counts in a watershed, and maybe some of the advantages of beginning to explain away some of those sources, those fecal sources, but then also talk just a little bit about other types of recreational water bodies, since Tillamook and some of the Great Lakes areas may be, they may be slightly different from others that others are working on.
Hi, thanks, Rachel. Yeah, so EPA method 1696 and 1697 are two qPCR-based human-associated molecular microbial source tracking tools that have been nationally validated by and standardized by the US EPA. So in terms of the question about water types, in my presentation, I gave some examples of us using them in freshwater systems, such as in Tillamook, Oregon, and in the Great Lakes. But one of the parts of the national validation effort for the US EPA was to actually test them in marine waters as well as estuarine waters. Tomorrow there is a presentation in session 4 by Amy Zimmer-Fost that's now at SQUIRP, where she used these methodologies in fresh and estuarine and marine waters. In addition, we have some larger scale efforts that were planned to be done this year, but have been moved back to the next year because of issues with the pandemic and just having the beaches being under normal conditions. Right.
That's great. I want to make sure that we have time to talk a little bit to Shannon and Laura. So I'm going to ask another question of you. Laura, I'm busy clicking screens because I've lost my screen that I was on, so excuse me. Laura, you described in your presentation, which I really enjoyed, because this has been an area of effort that I know that has been very valuable to folks for across the country in terms of cyanobacterial toxin signals matched with patterns of cyanobacterial really cells or nucleic acids. You looked at certain lakes in Missouri, Iowa, and Kansas, and you've kind of examined in your talk a range of technologies, but in the end, what, one of your slides, for example, was complex with trying to show some of the development of blooms using kind of an overlay. What are some of your thoughts on the technologies and tools that you think could be absolutely kind of taken and moved forward with, more immediately, and which things are a little bit more long-term?
Thanks for the question. I think that immediate, the Cyan Project has really provided a lot of useful data. I know our state of Kansas used that data a lot during the pandemic because they weren't able to do a lot of field work. So,
Laura, would you be able to hold your microphone kind of close to your mouth? It's going kind of in and out. If kind of hold it there.
Sorry, is that better?
That is better. Thank you.
Okay, sorry. Yeah, so the state of Kansas used the Cyan data a lot during the pandemic because they weren't able to do a lot of field work. So it was a really useful tool to them. And I think that a lot of the other states are going to implement using that satellite data to remotely And then long-term, we've worked again with the state of Kansas for their lab, developing these cyan detect and other molecular techniques to use for toxin analysis in their waterways. So they're working towards developing that method in their lab. Maybe that'll help. Is
that is helping. Thank you.
Okay. So the state of Kansas, we were able to do some training in their lab to get the molecular techniques up and running for them to use for cyanotoxin detection. And then we're working with a lot of our local parks and recs this summer to do some monitoring in some urban lakes using the profiler that I discussed, and then another project we've got working with ORD and Narragansett, where we're developing cheap sensors, and cheap is the acronym for a low-cost sensor panel, and not only temperature, which someone asked about, but also DO, conductivity, pH, trying to look at an early warning using a very inexpensive technology to give home associations and local lake managers, you know, the small scale people that don't have a lot of money to spend on expensive, kind of giving them the opportunity to have an early warning system.
Great, that's fantastic. I do want to shift to Shannon. You've got a couple of questions in the Q&A that are relevant to your presentation, and you're the last panelist that we haven't directly asked. I'm going to veer off road a little bit, Shannon, from the question that was originally provided to you, and just ask you a little bit more about the concept. You just kind of talked a little bit about, there are some places that qPCR just doesn't work, and there's some places that culture-based analysis just really isn't appropriate. So one question is really, do you see qPCR being kind of the gold standard, or moving away from culture-based methods and only using the culture-based methods in a small number of locations? Where do you see this going in the future, especially with Method C and the intercalibration work that you've completed and the digital PCR work that you're embarking on?
That's going to be very interesting. I seem to be about 10 years behind you and Julie Kinzelman and all the fun things you guys are working with. So there's going to be lots of methods available this whole time. And I think CoalAlert will always have a place because it's just foundational. And most of the states, their water quality standards are built on that. And for us in Michigan, it is a big deal to change a water quality standard. You know, it's like opening Pandora's box, and nobody wants to do that. So I would say that CoalAlert, culture-based methods are going to always have a foundational place for water quality standards. But for beaches, there's other things that we're considering, like, should I go swimming right now? I want to know right now. And also what Laura mentioned, these cyanotoxins, they are really causing people to scratch their heads, because if the wind changes, all the surface water goes to the other side of the lake, and now you've got a moving contamination source. And so those are some real weird things to consider what's going on. But, you know, with E. coli qPCR, and I know there's more E. coli methods out there, but we really landed on this one. We've been working with EPA, Rich Hoagland and Kevin Oshima for several years on developing this method. And it seems to have, we've got it working for like all of our beaches. We haven't found a beach that we couldn't get a signal, but we have found some beaches that we haven't found the source of contamination, and it's not following the best linear correlation with CoalAlert like all the other beaches. Does that make sense? Some are a 1-to-1 ratio. It's beautiful. It's perfect. Others, you'll see the qPCR shift a little bit in one direction. The other one, CoalAlert. And I say this by, once you have a linear correlation, then you also have to look at what are the exceedance rates. So in the presentation, I mentioned there's a 6% exceedance rate, and then there's a 10% exceedance rate. So we're looking at that, but I know we're running out of time. So I want to say this. Rich is working on this exact thing about all these different beaches, and it's with a regional applied research effort grant from EPA called a RARE grant. So if anybody's looking at how we're going to deal with all these little squirrely beaches that we haven't figured out yet, these qPCR methods work, the source tracking methods work, but they work in a different way that we wouldn't expect. Does that make sense? So they do work, but they're not following the same pattern as the majority of all the other beaches. So Rich is working on that. He's got several partners in the Great Lakes. So if anybody would like to reach out, and also all of our study was recently published in the Journal of Microbiological Methods. So what we don't cover here, it's in the paper. So thank you, Rich, and everybody for working on that.
Yeah, and big hopes for you with the digital PCR work as we close this panel, because that could potentially offer you the ability to, stop really comparing across culture and molecular methods so much. It might allow you to give the qPCR and a digital PCR an opportunity to kind of stand on their own, and the culture can still be valuable, but still stands on its own.
Yeah.
I just want to thank all of the panelists in this panel A, and thank everyone that's watching and listening for coming and for participating. We hope that maybe this could be the unique opportunity that we got in 2021, that maybe it doesn't have to be repeated, but thanks everyone for your participation and for your great questions.